Welcome to Biotanemy.
Bacterial transduction. Bacterial transduction is the process of transferring DNA from one bacterium to another by a virus named is a type of virus that infects and replicates within bacteria and archaea. They consist of a polyhedral head, collar, and helical tail. During transduction, the bacteriophage will attach itself to specific receptors called phage receptor binding protein, present on the surface of the bacterial cell. After adsorption, the phage muramidase will wither a small part of the bacterial cell wall, and will push its hollow core through it to inject DNA into the bacterial cell. On injection of DNA, the enzymes present in the phage DNA called early enzymes will start to synthesize the components for new virus particles using bacterial enzymes. During the replication, the phage enzymes will also weaken the bacterial cell wall. In the maturation phase, the head and tail proteins of bacteriophage will be assembled. Finally, the newly replicated bacteriophages will burst out from the bacterial cell. Fate of inserted phage genome. On insertion, the phage genome has two fates: either to follow the lytic cycle, or to undergo the lysogenic cycle and eventually go on to the lytic cycle. During the lytic cycle, all the previously discussed events will occur upon the phage takes control of bacterial cell machinery. During the lysogenic cycle, the phage genome will get incorporated into the bacterial chromosome. With the help of the CI repressor, the phage genome will prevent transcription and maintain lysogeny inside bacterial cell.
The bacterial chromosome carrying phage bacterium will replicate along with the chromosomal replication and continue to pass to other bacterial cells during bacterial division. The lytic cycle will be induced when some kind of stress happens to the bacterial cells. Based on the method through which the genome will replicated and packed inside the bacterial cell, transduction is again divided into three.
1. Generalized transduction. 2. Specialized transduction. And 3. Lateral transduction. 1. Generalized transduction. During generalized transduction, only bacterial DNA will be carried by bacteriophages. No viral DNA can be seen in the phage capsid. It may occur in two ways: recombination, or through head-full packaging. Head-full packaging is the mechanism through which bacterial DNA will get packed into the phage head, and the concatemer DNA or phage DNA is excised out.
2. Specialized transduction. Here, only a specialized set of bacterial genes will get transferred, which depends on where the phage genome will bind in the bacterial chromosome. When the lytic cycle is induced, the phage DNA will get excised along with some of the bacterial DNA adjacent to the phage DNA.
3. Lateral transduction. It is the process through which very long fragments of bacterial DNA will be transferred to another bacterium. It can transfer more genes and have higher frequencies compared to specialized and generalized transduction. They are specifically seen in Staphylococcus aureus. The prophage will start to replicate before they get excised from the bacterial chromosome, leading to the replication of adjacent bacterial DNA. The replicated DNA will be packed into the phage protein. Starting from its POC site, and adjacent bacterial genes will also occur in situ. Applications of bacteriophage. 1. As an antibiotic, since they can kill the bacterium. 2. In the food industry to obstruct food spoilage by bacteria. 3. Indicators of water contamination. 4. In diagnosis. 5. Hinder bioweapons and toxins like anthrax and botulism.
That's all for today's video. Thank you for listening. Hope all of you understand the concept, and for more queries and proposal, please comment down. See you all in a new topic.